types of clinical trials available? Where can I find a clinical trial? And how will this study help other brain tumor patients? And so to do that, I wanted to set a stage with a little bit of background information. And some of these slides have more stuff on them than I think we need to share, but hopefully there's some points that'll be helpful. So brain cancer, unfortunately, is a small percentage of the whole cancer pie, as we can see here. And as a result, industry, which does a lot of the clinical investigation, may not have a huge interest in our particular problem because at the end of the day, the market is not a whole lot of people. So we make up a small percentage of the total cancer pile. If we just look at the amount of patients with any type of brain tumor, unfortunately, it's a small percentage that are able to participate in a clinical trial. So from this slide, we can see that there's a small percentage overall, but also small percentage of patients participating in clinical research. Now, one of the things to bear in mind, and it's always a bit of a challenge, particularly with a group like this, there's a lot of different types of brain tumors. So we're mainly focused on the primary brain tumors, meaning they originate within the brain, not those that maybe start somewhere else in the body and spread to the brain. But even amongst the primary brain tumors, and we can see from this pie chart, there's a whole lot of different types, okay? Most of the ones that kind of fall into the malignant category I've got circled over here with red, and unfortunately that actually makes up a big percentage of the primary brain tumors. Now, if we just kind of look at brain tumors, because sometimes I think there's a question about, why does mine make me eligible for this trial and not this particular trial? And so ultimately we've got to understand this in the context of the biology of the disease. Here is an example of a type of tumor that arises from the lining of the brain, or maybe the lining of a cranial nerve, called a meningioma. We can see here's an example where the dura, this leathery lining of the brain, has been folded back. And we can see this tumor is really fairly circumscribed. It's fairly discrete. It causes trouble mainly by just pressing on the brain. Now, there are times meningiomas don't behave like that, but the vast majority of the time, these are fairly amenable to standard therapies. That is contrasted to glioma. Glioma's something that actually is within the substance of the brain. Even though we call this a solid cancer, it's really not a solid ball that the surgeon can just go in and shell out. To a degree, it's integrated and mixed into the brain. It carries a lot of biological features like proliferation and blood vessel formation, invasion, very resistant to even an environment that's inhospitable to normal cell growth. So it has a biology and a behavior that's very different from, say, a tumor like a meningioma. Now, excitement is occurring primarily related to a load of research that's understanding the biological origins of these diseases. Now, we're making a lot of headway in that aspect. That doesn't mean we understand why people get the disease, and we'll touch base on that in just a second. But this is also something we've got to keep in mind when we think about clinical trials. So a lot of times people can have a type of primary brain tumor called a glioblastoma multiforme, or a glioblastoma. But there are really two ways that this can occur. One way is it just shows up brand new. So we call that a primary or a de novo. Another way is this more stepwise, where it starts as a lower grade and then it goes to an intermediate grade and then it becomes a glioblastoma. And if we look at the molecular alterations, and there's no test on what all this means, but you can see the ones that lead to a primary glioblastoma, they differ from the pattern that we see over here in this more stepwise incremental progression. This side is usually younger patients, and this side is often an older patient. But we'll see a lot of clinical trials that'll talk about for glioblastoma, but if you fall into this category, what we'd call secondary, you may not be eligible. So there's a lot of little intricacies that may not be all that apparent to a patient or their family caregiver, when you're trying to search out this information. There are reasons for that. Some of these are due to the advances in our ability to do something called sequencing, meaning understanding the genetic sequence of a tumor and analyze that in a fashion with advanced bioinformatics and biostatistics. We have begun, and a lot of this led by the group at MD Anderson as well as multiple centers, but we really see that glioma is broken down into a lot of little pockets of tumors that share certain molecular features. They may be more likely to occur at certain ages and have different pathologies or histologies. And we're seeing this used increasingly to drive decisions related to clinical trials. Now, we need to have clinical trials, though. This category of tumors, kind of a median age of 56, men more likely than women, Caucasians more likely than African Americans. We've got standard treatment, but unfortunately standard treatment isn't really getting us where we want to be. There's some metrics that the NCI uses. One is called the years of life lost due to cancer. This is a measure of the burden of this disease on the individual, and it's high. Despite being a small piece of the pie, it carries a high burden for the patient and their families, really significant in children and young adults. And then something we all have to face. There's even if when you see me at UAB, you get this questionnaire. It wants to know if I discuss the economics of healthcare with you, which usually we don't get time to, but I think we have to be alert to that issue. But the public health cost of this disease is amongst the highest in oncology. A lot of that winds up getting borne by the family who is touched by this disease. Now, I wanted to briefly tell you a little bit about the etiology. So there's not anything we do that gives you a brain tumor that we know of. There's a few things that are pretty rare, like ionizing radiation for children. That goes back to how we sterilize the nervous system for children with leukemia. But for the most part, there's not something that we are doing, a decision that we're making that puts us at risk. Big studies called genome-wide association studies are beginning to identify what we call the risk alleles. These are the little variants in all of us that confer susceptibility to something. And we are making tremendous progress with that. This is a form of clinical research that's non-treatment related, but provides information that actually will wind up being incredibly important in better understanding who's at risk for the disease. By understanding what drives risk, it'll make it where we can better treat people with the disease. Now, I think we all are aware that there are three modalities we use: surgery, radiation, chemotherapy, all with pros and cons. I'm not going to detail that because I think you've heard some of that earlier. But now let's jump in a little bit about clinical trials. So kind of two things to be alert to. Clinical trials can be grouped based on the histopathology, meaning the type of tumor that the neuropathologist or the pathologist diagnoses you with. And so these are names like astrocytoma, glioblastoma, ependymoma, meningioma. Increasingly, we have clinical trials where patients are grouped irrespective of the cancer pathology, but more based on certain molecular alterations. Okay, so you could be in a clinical trial with somebody with breast cancer and you've got a glioblastoma, but it's because you share a common molecular alteration that is actionable. By actionable, I mean there's a drug available that blocks the effect of that mutation. These are called basket trials. So it's not grouping people based on pathology, it's grouping based on an actionable molecular alteration. There's a couple that are open right now. One is called the NCI MATCH trial, and then there's another run out of Dana-Farber that we're working to get open. It's called the INSIGHT trial. So again, these are not grouping people based on the pathology, but more on the mutations that are present in the tumor. Now, one of the things that I always go over with the fellows, the junior faculty, is, we can't be presenting clinical trials to patients if we're not willing to say what this is. So a clinical trial definition is a form of planned experiment that involves human participants and is designed to identify the most appropriate treatment for future patients with a clinical or some form of medical condition. So a lot of times we use a little more sterile terminology like clinical trial or study, but at the end of the day, at whatever level, it's some form of an experiment where we're trying to sort out if this is a benefit. I believe that at all levels there's a benefit to the participant, but we have to accept this definition. Now, this is something that I think a lot of people struggle with. What are these phases to clinical trials? And they're really kind of 4 phases. I got another slide that's got the first one. I don't know why it escaped me here. But you will often hear things about phase 1 studies, phase 2, or phase 3. Let's go to my other slide. It's a little better. It's got some information broken down. Increasingly, we're looking at something called phase 0 or pilot studies. A lot of times these studies, the goal of them is to prove some form of a biological principle. Examples of these in brain cancer patients may be you have a tumor, it didn't respond to the initial treatment, and we need to change treatments. We're going to go to a new type of therapy, but you would benefit from a surgery. Okay, so maybe we're going to give you this experimental drug for some period of time before you have surgery, and then at the time of surgery, the surgeon would save some of the tumor tissue and it would be analyzed for things like, did the drug get into the tumor, at a high enough concentration? Is the target of that drug present? And were we able to inactivate that? These types, and then after surgery, you would continue on that particular drug. These are usually small numbers of patients, unique situations, limited doses. Matter of fact, it's more common where this is a drug that's approved for another cancer or has another indication, but we don't know a lot about it relative to cancer in the nervous system. And so we're trying to get a better handle on, one, how well does this even get into the nervous system, and if it gets in, is it blocking the target that we intend? Phase 1 studies, I think there's a lot of anxiety about phase 1 studies. Even within the oncology literature, there was an editorial in the Journal of Clinical Oncology, which is kind of our premier oncology broadly journal, about the role of phase 1 trials, how to explain that to patients, what to tell them. These are often trials where we aren't certain of the right dose, okay? And that uncertainty could be because it's just not been given to a lot of patients. It could be because we just don't know in this particular disease, particularly for us when we're dealing with the nervous system. And so many times the goal is to try to figure out what the right dose of terms you may see are something called the MTD. That's the maximum tolerated dose. So oncology through the years always had the mindset, well, let's give somebody as much of something as we can. That may not necessarily be right, to be honest with you. We're increasingly now using a term called the optimal biological dose. So it may not be you need to take, the dose that is just below where it gives you troubles, that the biological activity is somewhere different. The starting doses for these will often come from animal studies. They can come from other human studies. And there is dose escalation between groups of patients. Depending on the design, that may be between every patient, or usually in little groups like 3 or 6, but not the same patient. The same patient doesn't get different doses. And then there could be, depending on the drug, large or small changes. Phase 2 is drugs have gotten through this process and we're beginning to think there's some benefit, but we need something that's a measure of how effective this drug is. Increasingly, the FDA requires phase 2 studies to be randomized. The physicians do not like that. Patients don't like that either. The randomization is not to not getting any treatment, it's just to getting the standard treatment. And so we hate those situations. Often these are smaller groups of patients, to be honest with you, and the dose that is used came from the phase 1 study. Lastly, which there are phase 3 studies. These are the big randomized, they can be what's called blinded and controlled studies. It's usually an industry that's sponsoring this. Rarely does the NCI reach this level. That company has met with the FDA, and this is what they have to do to get FDA approval. The dose that is being used is a standard dose. It's the dose that if it got approved, a standard therapy with less toxicity. We do have to have some way of deciding if the treatment is working, if it's doing anything. And the criteria that are used fall into 3 categories. One is a response, meaning we see an actual change in the size of the tumor. Sometimes there's a biomarker, like if you've got prostate cancer, you've heard of the PSA, so there's a blood test that can be followed. Unfortunately, with majority of brain cancers, it's hard to measure response. I know that's hard to believe, but a lot of times as treatment goes along, the interpretation of the MRI is increasingly challenged because treatment alters the appearance on the MRI. And so response is sometimes a difficult thing for us to have a lot of confidence in. Something that we feel more confident in is if we can measure time to some kind of event. So the time from when you started the drug to the time we think the tumor began to grow. Unfortunately, how long somebody lives, survival is the one the FDA requires for any type of approval. In addition, clinical benefits are increasingly recognized as acceptable metrics, and so these could be quality of life, or these could be stabilization or improvement in neurocognitive function. This is an example, it's an older example when Dr. Henson was probably back at Mass General, but it's called the McDonald criteria. We have a new set of criteria we call the RANO criteria, response assessment in neuro-oncology. But basically it's different categories that we would put the response in, and then there are different metrics that we use to make that determination. So I know a lot of times when you're on a clinical trial, you're meeting with the treating physician, and maybe the MRI looks a little worse, but you're clinically stable or better, and you've weaned off your steroids, and a lot of times we think that that's a positive, and we want to continue with that particular therapy. Now, I just threw this up so you could kind of get an idea of all the steps involved in this, and I'm sure there's going to be more, or there have been more that have been stuck in since I made this slide. But a lot of times we have to go through a process where we get somebody to sponsor a clinical trial. That could be a pharmaceutical company, it could be the National Cancer Institute, it could be the hospital like Piedmont. So sometimes the institution will sponsor some form of clinical research. Have to generate a protocol document and consent form. We got to try to get some money, usually from the NIH. There are different committees that have to approve clinical trials, and ultimately a committee called the Institutional Review Board. There's some of these that are professional that serve the country, but these are committees made up of not just physicians, but other people knowledgeable, patients and caregivers populate these committees and require approval before the protocol can actually open. Reports have to go to sponsoring agencies, and they have to be registered with something called clinicaltrials.gov. If you don't do that registration, there's a fine, and that fine is high. It's in the thousands of dollars. But as you can imagine, this government website is torture. So it's very difficult to kind of fill out all the information that they want. And so I usually just keep adding stuff until it accepts it. I'm trying to be honest, but usually sometimes you can't get some things to fit in there. All right, so sponsors of clinical trials. So sometimes you may hear a term investigator-initiated. That would be like if Dr. Dunbar had a project, she wanted to start a clinical trial, and Piedmont was going to sponsor it, and it would be an investigator-initiated study. A lot of studies are sponsored by the NIH. There are big cooperative groups across the country through the National Cancer Institute, so there are opportunities through that. The Department of Defense, through the Defense Health Program, these are the congressionally directed medical research program, also sponsor a lot of clinical trials, and then industry. The pharmaceutical industry or device manufacturers will sponsor a lot of clinical research. I want to show this as an example of a clinical trial. So for years we had a lot of discrepancies in how we treated people with low-grade gliomas. There was a lot of kind of things that could happen to you, and we didn't have a consistent way. You may get surgery followed by radiation, you may get surgery followed by chemotherapy, or you may get surgery and then both of them. And there was a lot of controversy about when you actually had to do this. And so this is an NCI. The SWOG is an entity, one of the National Clinical Trial Cooperative Groups, instituted this clinical trial. Ed Shaw started it back in the 1990s. It was reported in 2006, maybe 2014, I'm sorry, at the American Society of Clinical Oncology meeting. But if we look at this study, it put people into different groups. So there was a low risk just based on your age and how well the surgeon got the tumor out. And then a high risk, so if you were older than 40 or they couldn't get much of the tumor out. And then you would get randomized to 2 different treatments. And what we found, and you can see this took many years before this matured, but if you got the treatment that involved radiation therapy and chemotherapy together over radiation alone, then people did better. They lived longer. And so as a result, and this is just another way to look at it, this is a lot of times if you look at the literature on the internet and the medical journals, this is something called a Kaplan-Meier survival curve. And it's fairly easy to see that this is separated, and so that's a big benefit. Sometimes we can just look at the numbers. A lot of times I find that easy. That's easier to share with patients and families, so we can say, well, in this group they lived about 8 years, in this group about 13, or sometimes a percentage, like how many folks alive at 5 years can sometimes be another way of describing that benefit. And as a result of that study, this treatment got incorporated into our national guidelines. These are generated by something called the National Comprehensive Cancer Network, and so it altered the way treatment of low-grade gliomas occurred. So that clinical trial benefited patients, but also benefited all those patients to come now because we have a new type of treatment recommendation. All right, when to consider clinical trials. So the optimal time may be tumor type driven. And by that I mean a meningioma, standard therapy, very successful for the vast majority of patients. There is a percentage that it's not successful, but the majority with a meningioma, the standard therapy is going to, 1, be tolerated, and 2, lead to a high likelihood of curing you. Glioblastoma, unfortunately, standard therapy is limited. There are folks that are clearly doing well, but unfortunately we got to do better here. And we got to think about your patients, or you're thinking when you're newly diagnosed, do I do a clinical trial, or do I look at a clinical trial maybe if it comes back or at recurrence? So there are different windows in time spatially that we have to be alert to. There are important exclusion criteria to be aware of. Many times this is overlooked, but the number and the types of treatments that you've had to date can affect your eligibility for future clinical trials. So it's important to review that with your treating physician and know that if you do this, then I'm going to maintain eligibility for in the future. Now sometimes, there may not be a clinical trial. We may have to do a different treatment. We don't have a choice. We've got to move on. Sometimes surgery could impact eligibility. So a lot of clinical trials, particularly if it's a tumor growing back, it may require that there's a nodule of tumor that they can see, okay, meaning measurable disease. And so if there's not any, then that could impact eligibility. And then there's got to be a window of time where the toxicities from your prior treatment have resolved. So like if you're taking the drug Timodar and you took it and for some reason had trouble, got an MRI the day after, well, you can't start a clinical trial for 4 weeks after you finish that dose of Timodar. So something to be kind of alert to. Where do you get information about clinical trials? So obviously, we all get information about everything from the internet. So when my refrigerator every morning I would go, it had this puddle of water by the right front door. You can type that into Google and it will tell you the things to look at in your refrigerator. And I fixed it. It was the thing that defrosts the drain was filled with ice. But it took so you can learn a lot from the internet, but be wary of just, a Google search and what you may get. I think it's best for people to go to the foundations. Obviously, ABTA, Musella provide a lot of useful objective information relative to clinical trials for patients with brain cancers. And then also clinicaltrials.gov is another source. And this will actually give you emails to the investigators. That's how the lady from Oregon who emailed me this morning had gotten me off clinicaltrials.gov from a trial we had. This, though, is a little more laborious maybe in understanding. These, I think, are a little more helpful as far as figuring out what is timely for you. All right, so academic medical centers, so those with comprehensive or clinical cancer centers, large community hospitals with a cancer center, a big oncology practice. So going to those sources, particularly in your community, is a very important thing. Probably the best, to be honest with you, patients and families. I know this is a little bit of a challenge because the frequency of this disease is not that high. I basically only do email. We don't have Facebook. We don't have Twitter at UAB. So I don't really, I'm socially isolated electronically, but a lot of information can be extracted from other patients and families. Now, things to consider relative to risk. So alert to that. That can really be a factor. The consent process is when the treating physician or representative is getting you to sign some document that says you're willing to do this. If you've ever done it, some of these documents 20, 30 pages long. There's no way you and I can go through all this within the time frame of a clinic visit. So often it needs to be provided to you well in advance. You need to have time to take that home. I have people highlight on it, write on it, and then we'll come back and discuss it and sign that document. It's not a legal document, so it's not like you're doing your mortgage. It's a document that says we have discussed this and we're both comfortable. Reimbursement. So often there is reimbursement for travel. If you're a long way away, you need to stay in the hotel so you can get all the issues addressed. Those are options. How long will I be on this clinical trial? Usually there's 3 reasons to come off. 1 is it's not working, so you wouldn't want to stay on it. 2 is it's got some kind of toxicity or side effect that's worse than the underlying disease or taking away your quality of life. Or 3, you've just been on it for some period of time and you just want to stop. So just voluntarily want to stop. Had a lady who came to see me from Mississippi, and she had a recurrent glioblastoma, went on a phase 1 clinical trial and had a dramatic response. And she stayed on this drug for 4 years, seeing me every month. And neither she nor I we couldn't get the courage to just stop. Finally we did. But we were always afraid if we stopped and then the next time she came back, the MRI changed, we would blame it on doing that. So there's always the voluntary side. So just to conclude, 4 points. 1, brain cancer is a significant health problem. The burden related to neurological disability is tremendous for patients, and this affects their caregiver and their community. Advances in the understanding of the molecular events that promote tumor behavior, these are the things that cause the problems for patients, morbidity and mortality. That is being understood. There are non-therapeutic ways to participate in that, and those are helping us develop new treatments. Patient participation in clinical trials is critical. It's the only way we're going to make some advances. and alleviate the burden of this disease. Factors to consider, we've talked about a lot of things, but types of trials, what the endpoint of the trial is, if it's feasible for you and your family, and what the cost may be. And